Good morning, everyone, and welcome to today's presentation. I'm excited to introduce our topic, Explanation of Taxonomic Distinctness and Variation. Today, we'll explore how these concepts help us understand the evolutionary relationships within ecological communities. We'll look at what taxonomic distinctness is, how it's measured, and what its variation tells us about biodiversity. So let's get started and dive into these fascinating ideas that go beyond simple species counts to give us a deeper insight into the natural world. Now, let's take a closer look at taxonomic distinctness, often represented as delta asterisk. This measure helps us understand how evolutionarily diverse a community is. An alternative way to formulate it involves considering all the species in the community, which we denote as S essentially. S represents the total number of species present. When we calculate delta asterisk, we typically use the number of unique pairs of species, which is S times S minus 1, divided by 2. This value serves as our denominator and helps standardize our measure. Using this approach allows us to compare different communities more fairly, even when they have varying numbers of species. The main purpose here is to make meaningful comparisons across communities that differ in species richness. Without this standardization, communities with more species might naturally seem more diverse, but that wouldn't necessarily reflect true evolutionary differences. Finally, interpreting delta asterisk values is quite straightforward. Higher values suggest that, on average, species in the community are more evolutionarily separated from each other. This indicates a greater evolutionary diversity within that community, which is a key aspect of biodiversity we're trying to assess. So overall, this measure gives us a standardized way to compare how evolutionarily distinct communities are, regardless of how many species they contain. Now, let's take a closer look at what we mean by variation in distinctness, which we denote as lambda plus. Essentially, this measure helps us understand how uneven the relationships are among different species in a group. When the variation is high, it indicates that some species are very closely related. For example, they might belong to the same genus, while others are much more distant, like species from different phyla. On the other hand, low variation means that most pairs of species have similar taxonomic distances, suggesting a more uniform level of relatedness across the board. To quantify this, we use a specific formula that considers the distances between pairs of species. Here, dig represents the taxonomic distance between species I and J. When we calculate this for all unique pairs, we get a comprehensive view of the overall variation in the group. What's important to note is that both equations we use, considering all pairwise distances or just the unique pairs, will lead to the same result. This consistency reinforces the robustness of the measure. By understanding this variation, we gain insight into the structure of biodiversity within a group, which ties back to our broader goal of assessing how species are related and how diverse the group truly is. Now, let's take a look at what the structure of a community can tell us about its underlying dynamics. When we see a high delta asterisk value, it indicates that the community spans a broad range of taxonomic levels. That means we're likely observing species from many different families or even phyla, suggesting a very diverse and potentially functionally broad community. On the other hand, a low delta asterisk points to a community dominated by closely related species, often within a single family. This kind of structure suggests more specialized interactions or perhaps a community that has evolved within a more narrow ecological niche. Moving on to the metrics of similarity, a high lambda plus value, which we call hierarchical clustering, shows us that the community contains both very similar and very dissimilar species pairs. This indicates a complex structure with a mix of close relatives and more distantly related species, reflecting layered relationships within the community. Conversely, a low lambda plus suggests that relationships are more evenly distributed. In other words, species pairs tend to be neither very similar nor very dissimilar, indicating a more uniform pattern of relatedness across the community. Overall, these metrics, delta and lambda plus, are crucial for understanding the community's structure. They help us see whether it's dominated by a few closely related species or spans many levels of diversity, which in turn informs us about the ecological processes shaping that community. Connecting this to our broader theme, these insights allow us to better grasp how community composition influences ecosystem function and resilience. Now, let's take a look at what we call conservation insights. When a community has a high delta star value and a high lambda plus value, it indicates something quite important. Essentially, this means the community contains unique evolutionary lineages, species that are distantly related, and also has redundancy, meaning there are closely related species clustered together. This combination is actually very beneficial. It provides resilience against environmental changes because those unique lineages add distinctiveness, helping the community withstand disturbances. In other words, these communities are both diverse in their evolutionary history and robust in their structure, which is exactly what we want when thinking about conservation. On the other hand, when we see low delta star and low lambda plus values, the picture changes. Here, the community tends to be more homogeneous, with less variety in evolutionary lineages and fewer closely related species clustered together. This kind of community is more vulnerable to disturbances because it lacks that diversity and redundancy that can buffer against change. So, in the bigger picture, understanding these metrics helps us identify which communities are more resilient and which might need extra protection. It really ties back to our overall goal, preserving not just species, but the evolutionary richness and resilience that make ecosystems sustainable over time. Now, let's look at some practical examples to really clarify these concepts. What we'll do is walk through how to calculate taxonomic distinctness, which we denote as delta star, and the variation in taxonomic distinctness, known as lambda plus. These calculations are based on real species data, so you can see how they work in practice. By applying these examples, I hope it becomes clearer how these metrics help us understand the diversity within a community, not just in terms of species count, but also considering their evolutionary relationships. This connection to the overall theme of biodiversity assessment is really important because it gives us a more nuanced picture of ecological health. So, as we go through these examples, keep in mind how these calculations can be applied to your own data or research questions. Now, let's walk through how we calculate taxonomic distinctness, or delta asterisk, using a simple example with 8 hypothetical towns. Imagine these towns are organized hierarchically, much like how towns are grouped into prefectures, regions, and countries. This hierarchy reflects their evolutionary relationships, so shared higher levels, like the same region, suggest they are more closely related than just sharing a country. Since we don't have real data on the exact differences between taxonomic levels, we assume each step in this hierarchy is equal, say, 1 kilometer for simplicity. This uniform distance helps us measure relationships in a neutral way, giving equal weight to each level of taxonomic separation. Let's look at some specific examples. First, towns within the same prefecture, think of these as being in the same genus, are the most closely related. The distance between them is just 2 units because they share a common immediate grouping. For example, towns A1 and A2 are both in prefecture A. To get from A1 to A2, you go from A1 to the prefecture level and then down to A2, totaling 2 units of distance. Next, consider towns in different prefectures but within the same region, similar to being in the same family or order. Here, the distance increases to 4 units, since you need to go up from each town to their shared broader grouping, the region, and then down to the other prefecture. For instance, A1 in prefecture A and B1 in prefecture B, both in region 1, have this intermediate distance. Finally, towns in entirely different regions, like belonging to different phyla, are the most distantly related in this hierarchy. Their distance sums up to 6 units because you have to traverse multiple levels, from the town up through the prefecture, region, country, and then down through another region and prefecture to the other town. For example, A1 and C1 are in different regions, making their taxonomic distance 6 units. Across all 8 towns, we can form 28 unique pairs, and by summing the distances for each pair, we get an overall measure of taxonomic diversity. This example helps us understand how the distances reflect evolutionary relationships, which is central to calculating the Taxonomic Distinctness Index. This method enables us to compare biodiversity in a way that accounts for how related different species, or towns, are within a hierarchical framework. Now, let's take a closer look at how we calculate taxonomic distinctness, specifically the delta asterisk index, through this example. As you can see here, we're focusing on understanding how different species relate to each other within a taxonomic tree. The core idea is to measure the average taxonomic distance between all pairs of species in a sample. This helps us assess biodiversity in a way that accounts for the relatedness of the species, not just their presence or absence. In this example, we've identified the pairs of species, and for each pair, we determine their taxonomic distance by looking at their position in the hierarchy, like genus, family, order, and so on. Then, we sum these distances and divide by the total number of pairs to get the average. This average, the delta asterisk value, gives us a sense of how taxonomically diverse our sample is. Understanding this calculation is crucial because it allows us to compare biodiversity across different communities or ecosystems in a more meaningful way, considering not just how many species there are, but how different they are from each other taxonomically. This example illustrates that process step by step, making it clearer how this metric can be a powerful tool in our biodiversity assessments. Now, let's take a closer look at how we calculate taxonomic distinctness, specifically delta star, using this example with these 8 towns. First, we identify all the possible pairs between these towns. In this case, there are a total of 28 unique pairs. Next, for each of these pairs, we determine their taxonomic distance based on how closely related they are within the hierarchy. This involves looking at whether they are in the same prefecture, the same region but different prefectures, or entirely different regions. The table here breaks down these relationships. For example, when 2 towns are in the same prefecture, like A1 and A2, the distance is quite small, say 2 units. If they are in different prefectures, but within the same region, like A1 and B1, the distance increases to 4 units, since they're a bit more taxonomically distant. And finally, for towns that are in different regions altogether, such as A1 and C1, the distance jumps further to 6 units, reflecting a greater taxonomic separation. Adding up all these individual distances for each pair gives us a total of 136 units. This total helps us quantify how taxonomically diverse this set of towns is, which is central to understanding biodiversity patterns in our broader analysis. So, by breaking down the pairs and their relationships, we can systematically measure how distinct or similar these towns are within the taxonomic hierarchy. In this slide, we're looking at an example of how we calculate taxonomic distinctness, specifically the delta asterisk value. So imagine we have data for 8 different species. When we look at all the possible pairs between these species, there are 28 unique pairs in total. This is calculated using the formula S times S minus 1 divided by 2, which for 8 species is 8 times 7 divided by 2, giving us 28 pairs. Now, for each of these pairs, we determine their taxonomic distance, which is based on how closely related they are within the hierarchical classification system, like genus, family, order, and so on. When we sum all these distances across all pairs, we end up with a total of 136 units. This total reflects the overall taxonomic diversity in our sample. It's important to note that in most ecological analyses, we use this same denominator, S times S minus 1 over 2, to normalize our calculations. This approach makes sense because it accounts for all the unique pairs without double counting. Finally, to find the average taxonomic path length, or delta asterisk, we divide the total sum of distances, 136 units, by the number of pairs, which is 28. This gives us an average value, our taxonomic distinctness, that represents how taxonomically diverse our sample is on a per-pair basis. Overall, this measure helps us understand the structural diversity within a community, and it connects directly to the broader theme of assessing biodiversity in ecological studies. Now, let's take a closer look at how we calculate taxonomic distinctness, specifically the delta asterisk value, using an example with 4 species. Imagine we have these 4 species arranged within a taxonomic tree, which shows their evolutionary relationships and classification levels. The key idea here is that taxonomic distinctness helps us measure how evolutionarily diverse a set of species is, based on their taxonomic hierarchy. So, in this example, we look at all possible pairs of species within our sample, and for each pair, we determine how taxonomically related they are. This involves considering the taxonomic levels they share, like genus, family, order, and quantifying that relationship. The delta asterisk calculation then takes the average of these pairwise taxonomic distances. By doing this, we're essentially capturing the overall taxonomic breadth of our sample, which is especially useful for understanding biodiversity in ecological studies. Remember, the higher the delta asterisk value, the more taxonomically diverse our sample is, indicating a broader range of evolutionary lineages. This example illustrates how we translate the structure of the taxonomic tree into a meaningful numerical value, helping us compare biodiversity across different samples or environments. Now, let's walk through how we calculate taxonomic distinctness with this example involving 4 species. First, we assign path lengths based on their taxonomic hierarchy. So, when we're talking about the same species, the path length is 0 because they are identical. If they belong to the same genus but are different species, the path length is 2 steps, going from one species up to the genus, then down to the other species. Moving further up, if species are in different genera but within the same family, that's 4 steps. Species to genus, genus to family, then back down to the other genus and species. And finally, if they are from different families altogether, that's 6 steps, traversing all those taxonomic levels. Next, we create our distance matrix, which shows the pairwise distances between each species. For example, species A and B are 2 steps apart because they share the same genus, but species A and D are 4 steps apart because they belong to different genera within the same family, or even different families, depending on the hierarchy. Then, we sum all these pairwise distances. Remember, each comparison is counted twice, once in each direction, so we add up all those values. When we total everything, the sum comes to 40. This total helps us understand the overall taxonomic diversity within this sample, giving us a quantitative measure of how taxonomically distinct these species are from each other. This example illustrates the practical process of calculating taxonomic distinctness, which is central to evaluating biodiversity at a higher taxonomic level. It's a useful way to capture the evolutionary relationships and diversity within a community. Now, let's walk through how we calculate the taxonomic distinctness, specifically the delta asterisk value, with this example. We start with our sample data, which includes 4 species. Our goal here is to find the average taxonomic distance among these species, which tells us about the overall diversity. First, we set S, the number of species, to 4. Next, we determine the total number of pairs we can form from these species. Since each pair involves 2 different species, we calculate S times S minus 1, which gives us 4 times 3, totaling 12 pairs. We exclude self-comparisons because comparing a species to itself doesn't add any new information, so that leaves us with 12 pairs. Now, for the first method, we divide the total sum of taxonomic distances, which in this case is 40, by the number of pairs, 12. This results in approximately 3.33, giving us the average taxonomic distance per pair. Alternatively, if we only count each unique pair once, we divide by the number of unique pairs, which is S times S minus 1, divided by 2. That's 4 times 3 divided by 2, resulting in 6 pairs. The list of these unique pairs is AB, AC, AD, BC, BD, and CD. When we sum their specific taxonomic distances, which are 2, 4, 4, 4, 4, and 2 respectively, the total adds up to 20. Dividing this total by the number of unique pairs, 6, gives us 20 divided by 6, which again is approximately 3.33. And notice, both methods, whether we consider all pairs or just the unique ones, yield the same delta asterisk value. This consistency confirms our calculations and highlights how delta asterisk provides a meaningful measure of taxonomic diversity within our sample. Now, let's talk about variation in taxonomic distinctness, represented as lambda plus or lambda plus. This measure helps us understand how much the genetic or evolutionary distances between species in a community vary from the average. Essentially, it tells us whether the relationships among species are fairly consistent or if there's a lot of diversity in how closely related they are. When lambda plus is low, it indicates that the species in the community tend to have similar levels of relatedness, meaning their evolutionary distances don't vary much. On the other hand, a high lambda plus value suggests there's greater diversity. Some species might be very closely related, while others are quite distant. This is important because it gives us insight into the community's structure and diversity. If we're seeing a high variation, it might mean the community includes a mix of closely related and more distantly related species, which could have implications for how the community functions or responds to changes. So, in the context of our overall analysis, understanding this variation helps us grasp the complexity of the community's evolutionary relationships, beyond just counting species or looking at their abundance. It's a key piece in understanding the depth and breadth of biodiversity within the ecosystem. Now, let's look at how we calculate the variation in taxonomic distinctness, specifically using this example. First, we start by calculating the mean distinctness, which is given as 3.33. This value represents the average taxonomic distance across all pairs in our dataset. Next, for each pair of taxa, we find the deviation from this mean. For example, the pair A to B has a value of 2, so we subtract the mean, 3.33, resulting in minus 1.33. We then square this deviation, which gives us 1.78. This squaring process is important because it ensures that deviations in either direction, above or below the mean, contribute positively to the overall variability. We repeat this process for all pairs. For instance, A to C has a value of 4, so the deviation is 0.67, and squared, that's 0.44. Similarly, A to D also results in a deviation of 0.67 and a squared value of 0.44. When we look at pairs like B to A or C to D, we see deviations of minus 1.33 or minus 1.33 respectively, with squared values of 1.78 each. These squared deviations are crucial because they quantify how much each pair's taxonomic distance deviates from the average, contributing to the overall measure of variability. By calculating these squared deviations for all pairs, we can assess the degree of taxonomic variation within our community. This approach helps us understand the heterogeneity of the community's taxonomy and how evenly or unevenly related the taxa are overall. This step-by-step process ultimately allows us to compare different communities or samples to see how their taxonomic structures differ, which is a key aspect of understanding biodiversity patterns. Now, let's look at what variation in taxonomic distinctness, or lambda plus, actually tells us. Essentially, it measures how much the genetic or evolutionary distances between species in a community differ from the average. So, if lambda plus is low, it means the relationships between species are quite consistent. Most species are similarly related. Conversely, a high value indicates there's more diversity in how related the species are. In our calculation, we first summed the squared deviations. We did this by multiplying 4 by 1.78 and 8 by 0.44, which gives us 7.12 and 3.52 respectively, and adding those up to get a total of 10.64. Next, we divide this sum by the total number of comparisons, which is 12, to find lambda plus. That gives us 10.64 divided by 12, resulting in a value of approximately 0.89. What does this number mean? Well, looking at the interpretation, the average taxonomic distinctness, or delta star, is 3.33, which tells us about the overall relatedness in the community. And lambda plus at 0.89 indicates that the pairwise distances are fairly consistent around this mean. Most species pairs are either closely related, like within the same genus, or more distantly related, like between different genera. So, in summary, this relatively low lambda plus value suggests that there's not a huge amount of variability in the relatedness among the species in this community. It points to a fairly structured and predictable pattern of relationships, which is important for understanding the community's overall diversity and evolutionary history. Now let's look at this final example, which involves multiple phyla. The goal here is to understand how we calculate taxonomic distinctness, or delta asterisk, and its variation, lambda plus. We're working with 4 marine species from 3 different phyla. What's really interesting is how we assign path lengths based on their taxonomic hierarchy. For example, if 2 species are the same, we exclude that from our calculations. When they're from the same genus but different species, the path length is just 1. If they're from different species but within the same genus, the path length is 2, which includes moving up to the genus and back down. Going further, if they're from different families but within the same order, the path length increases to 3. And as we move up the hierarchy, if species are from different orders within the same class, the path length is 4. When they're from different classes within the same phylum, it's 5, and finally, if they're from different phyla altogether, the path length reaches 6. This example highlights an important point. Even if the average taxonomic distinctness is high, meaning species are generally far apart, it's possible to have low variation if most pairs are similarly distantly related. This kind of detailed breakdown helps us see not just how distinct species are on average, but also how consistent or variable those relationships are across our sample. It's a powerful way to understand biodiversity at different taxonomic levels. Now let's look at this final example, which involves multiple phyla. Here, we're demonstrating how to calculate 2 key metrics, taxonomic distinctness, or delta asterisk, and its variation, denoted as lambda plus. We have 4 different marine species from 3 separate phyla, and the goal is to understand how taxonomic relationships influence these calculations. What's important here is how we assign path lengths based on the taxonomic hierarchy. Essentially, the more distantly related 2 species are, the longer their path length or pairwise distance. For example, when we compare Asterias, which is a starfish, to Carcinus, a crab, they belong to different phyla, so their pairwise distance is 12. Similarly, comparing Asterias to Littorina, which is a snail, also yields a distance of 12 because they're from different phyla. The same applies when we compare Asterias to Mytilus, a mussel, again a different phylum, so the distance remains 12. Now, looking at Carcinus versus Littorina and Carcinus versus Mytilus, these pairs also have a distance of 12, since they're from different phyla. The only pair that differs is Littorina versus Mytilus, which are both mollusks, but from different classes. This pair has a distance of 10, reflecting their closer relationship compared to the other pairs. What this example shows us is that even if most species pairs are quite distantly related, resulting in high average taxonomic distinctness, the variation, or how much these distances fluctuate, is low when most pairs are similarly distant. This highlights an important concept. High average values don't always mean high variability, especially when the relationships are consistently distant. Overall, this example emphasizes how taxonomy shapes the pairwise distances and helps us understand biodiversity patterns across multiple levels of life. It's a practical illustration of how these calculations reveal the structure of biological communities. Now let's look at this final example that involves multiple phyla. Here, we're calculating 2 important metrics, taxonomic distinctness, or delta asterisk, and its variance, called lambda plus. We're working with 4 marine species from 3 different phyla to see how their taxonomic relationships influence these measures. First, the key idea is that we assign path lengths based on their position in the taxonomic hierarchy. Think of it as measuring how far apart species are on the tree of life. When we calculate pairwise distances, most of these pairs are quite distant because they belong to different phyla, which results in a high average path length, or delta asterisk. Now let's move on to the variation, lambda plus. To find this, we look at how much the individual pairwise distances deviate from that average. We start by calculating the squared deviations for each pair. For example, for most pairs, the difference from the average is small, around 0.111, which means their distances are very similar. But there's one pair with a shorter distance, which slightly increases the overall variation. Adding up all those squared deviations gives us 3.333, then dividing by the total number of pairs, 6, results in a lambda plus of about 0.556. This relatively low number indicates that, although the average distance is high, the distances don't vary that much across most pairs. So to sum up, we see a situation where most species are quite distantly related, hence the high delta asterisk. But because many pairs share similar distances, the variation, or lambda plus, remains low. This example neatly shows how taxonomic structure can lead to high average distinctness, but still have low variability among pairs. It's a good reminder that these measures help us understand not just how different species are, but also how consistent those differences are across a community.